So today I will talk about communication in science, and the presentation is mainly directed at clinicians, but of course I know that there's always many patient representatives attending as well, so a lot of this will hopefully also make sense to you. So first, my disclosures. They are not related to this talk, so I will skip this slide, but during my next talk on Givinostat, I will go over them because then they are relevant. So what I will talk about today is how scientists communicate, how they communicate to each other, how does this work? And of course, we're in a therapeutic environment, so we mainly will discuss how scientists discuss therapeutic efforts. And of course, when you develop a therapy, you don't immediately start in the patient. You start in cell and animal models, and then eventually you move to clinical trials. And all these different steps are important because there's things in cell and animal models that you cannot do in a patient. You can, for instance, in a mouse model, you can study all the different muscles. You cannot take a biopsy from all the different muscle in a patient. But you have to bear in mind that these model systems are not perfect. So the next step is always more complicated. And that means that if there's success early on, there's no success or no guarantee for success in the subsequent steps. So just to make it more concrete, if something works in a cell model or in an animal model, that does not mean it will also work in a patient. Of course, we hope it will work in a patient, but we have to test this first. And that is what we do in the clinical trials. And then also what we often say when a clinical trial shows that a treatment that we tested doesn't work, we will say the clinical trial failed. Well, that is not a correct way to say it. The clinical trial is an experiment in humans that tests whether a treatment that showed promise in an animal model, whether that worked or not in humans. And if the answer is it does not work, then the trial is a success. Even though, of course, we would have liked another result, we would have liked the treatment to work, but the trial is done to see whether the treatment works in a human setting, and then the answer can be yes or no. So a failed trial is a trial where at the end of the trial, you cannot say whether the treatment works yes or no. That is a truly failed trial. So now back to communication, because of course, when we communicate, there is a sender and a receiver. And I can say something and I can mean something, but the way that you as the audience perceive it can be different than from how I intended it. And then I can say, you don't understand, but I mean, it's not your problem that you perceive things differently than the way I intended. So this is important. There's two partners when we communicate. There's a sender and a receiver. And I would like to get your mobile phone and to scan this QR code, and then to tell me what you think of when I say Barcelona. And there's no right or wrong answer, just when I say Barcelona, what is the first thing that pops up in your head? And then I will test it myself to see if it works. Oh, it works because someone already said city, opera, sun. I will add the Sagrada Familia, because that is where I am thinking of. So Gaudí. So what we see is, well, even though there's some words that more people think of than others, there's a lot of different things that people think of, and these are not right or wrong. This is just what you think of first when you think of Barcelona. And this is very important to bear in mind, that different people think of different things, because that is also how normal communication works. When I say something, because some words can mean different things. When I say Hamilton, you can think of the US founding father, you can think of the Formula One driver, you can think of a city, there's different things, you can think of the musical, there's different things, and it's one word. So this is important, that context is important, and the context is in what I tell you, but the context is also what is in your head because of your own personal experience. So basic communication. So how do scientists communicate? Of course, we also talk, but mainly what we do, we publish. We publish in peer-reviewed papers. And I think the clinicians, the researchers in the audience will know this. You submit it, your peers, your fellow researchers or clinicians will review it, will see whether it's scientifically sound, will come back with feedback. You then have to adapt it and resubmit. And in the end, when it's okay, it will be published. And it's good to bear in mind that there's two types of journals, scientific journals. There's journals with good standing, good reputations that do a rigorous peer review. But when you publish, as a researcher, you have to pay the publication fee to the journal, to the publisher. And so this is a business model for some publishers where they think, well, we don't really care whether the stuff is scientifically sound. We just want as many publications as we can, because then the researchers pay and we make money. And then, of course, the win for the researchers is that they have publication which looks good on their CV. The downside for the scientific field is, of course, that this is not well done science. So this is poorly done science. So when something is published, it really depends on which journal it's published in. Another way to communicate is at conferences, and then at a scientific conference, the peer review is done by the audience. And this is why, if you're as a patient representative, if you go to a scientific conference, you will see that the scientists try to convince the audience. They show in 5 different ways why things are the way they think they are. This is because they are used to convince things. And of course, in a patient conference, then the level of content needs to be easier, because patients don't know the jargon, they don't know the tools and the techniques usually. So this is how scientists communicate. But of course, when we talk about therapy, there's also companies involved, and mainly they do the clinical trials. They sponsor the clinical trials. And how they communicate, usually the first thing that happens when there's trial results is a press release. And this is not peer reviewed. So this is what how the company interprets the data. Very often there will be a quote of an academic, but of course the company is not going to quote me if I say, well, I think this trial shows that this really doesn't work. No, they're going to look for another academic then who says this is great. So they can choose who to use for this. Then later, there very often will also be publications in the scientific journal about the clinical trial, and then you can really go into the data and look what was done. And this is peer-reviewed, and also academic authors are involved. So this is, say, a higher level of evidence because more people critically looked at it. Also at conferences, there can be presentations on clinical trials. Very often this is done by academic presenters, but bear in mind that what they present is agreed upon content. So the companies have checked that the slides that the presenters present, that they are okay with it. So again, they're not going to select a presenter who's not that positive about their work. They're going to select someone who is positive, so they can influence it in that way. What is good to remember is that scientists are humans, and humans can make mistakes. Also, scientists are usually workaholics and very passionate about science in general, and even more about their own science. So they are enthusiastic and they may oversell just because they are so happy about the work that they're doing. They're also not trained to present to lay audience. So I mentioned already, very often when scientists present to patients, they will use a lot of jargon, and it's very easy to overestimate the level of knowledge of the audience. And well, for the clinicians and the researchers in the virtual room, we studied decades to come to where we are. So we forget that the level of knowledge that most people have is what they learned in high school, which can be 10, 20, 30 more years ago. So that's really a different level. So really it's important to do this at a lower level. But it's also good. So scientists are trained to be critical, but again, scientists, we are humans. So we're often much more critical of others than of ourselves. So this is also good to know. So I have now a few examples of how science was poorly communicated. And this is from my paper a day thing, where every day I read a scientific paper. I will start again tomorrow after my holiday, and I will summarize, and if needed, I will criticize it as well. And on the World Duchenne Organization website, there's relevant threads about papers related to Duchenne. And this also means that I have some ideas of mistakes and misassumptions. And I will show a few brief ones to you now so that there's time for discussion. If we have time after my second talk, we can fill it up with a larger example. If there's no time, then we just skip that. That is not a problem. So again, be prepared because we're going to get interactive again. We have a mouse study. We have the MDX mouse that doesn't make dystrophin, that has a defect in the diaphragm function. And now we have treated it with something in blue, and you see that that something improves the diaphragm function. So this is mouse. But my question to you is, does this treatment work in mouse? And then again, you can scan and answer. It doesn't work at all. It works a little bit. It works quite well. It is amazing. I don't know how well it works. So I'm gonna wait again for you to provide your answers. I'm gonna give you 10 more seconds. And again, it's anonymous, so don't worry about getting it wrong. And I think, well, the majority seems to lean towards, I don't know how well it works. And actually, I think that is the best answer, because all you saw was a mouse without dystrophin and a treated mouse. What you didn't know was the wild type mouse, the function of the mouse with dystrophin. And that is now shown in this graph. And what you can see is that the diaphragm function is much, much, much better in the healthy mouse. And then this difference that you saw 2 slides back, that looked quite impressive if you ignored how the healthy mouse was doing, now is really quite trivial. So the people said it works a little bit. Well, maybe it works a little bit, but I would say it's really not worth studying in more detail because of this. And this is what you very often see, that people don't include the healthy mouse, and then of course you don't know how to interpret results. So other things to be aware of when you read scientific papers as a clinician, as a patient representative. Scientists can focus so much on their model system that they see some pathological aspect that is present in the model, but that's not present or not relevant in the patient. And then they start developing a treatment for this aspect. But well, if patients don't have this, or if patients are not bothered by it, how relevant is this as a treatment? So that is important to bear in mind. And then another thing is that, of course, scientists have publications, and once it's published in a high standing paper like Science, which is my next example, then that paper is good quality. But then institutes can also have a press release, and that can be picked up by the media, and then things can suddenly lead into a hype. So my example here is a dog model that was treated with CRISPR. So this was one dog where they did CRISPR treatment, and what they did was they restored a Becker type of dystrophin in one dog. They didn't do anything else, but the authors were very honest about this. A lot is yet to be done, but we show that in principle, in a dog, we can restore this Becker dystrophin in a Duchenne dog model. So that was what they said. And then you can see here what happened in the media. First, it was CRISPR fixes muscular dystrophy. Well, it didn't really fix it. It just restored a partially functional protein. At least this one said human trials are far off. A few hours later, muscular dystrophy halted by CRISPR. Are humans next? Well, it wasn't halted. They didn't even look in the function of the dog. So how did they know that the muscular dystrophy was halted? They only looked where the dystrophin was restored. And the author said, well, we don't know if this will ever reach humans. And then again, the next day, CRISPR successfully stopped Duchenne muscular dystrophy in dogs, and human trials are next. So this was a publication from 2018, and well, I know there's some trials that are happening in China, but there's no concrete CRISPR trials planned in the Western world in Duchenne, and also there's currently no evidence that this will be safe and effective and efficient. So just to show you how quickly the media can blow things up. Then a last example is drug repurposing, and that is when you use a drug that's been approved for one disease to treat, in this case, Duchenne. But there's things to consider, because if this drug was approved for an infection or for cancer, where the treatment would be only weeks or months, for Duchenne now suddenly the treatment is permanent. So that, of course, has much different impacts with the side effect profile, for instance. Also, if the drug was approved for elderly populations and now you want to treat it for children or use it for children, you probably will need to do more work. And the route administration may be a problem, because if it's really big pills, well, Duchenne patients might not be able to swallow this. And then very often what they start with is they should use this in animal model, and often they use a very high dose. And then the question is, well, the dose that they use in the animal model, can you use this in the clinic? And if the answer is no, then yeah, how relevant is this work? And then the last thing is, will this be safe? The fact that it's approved for one disease does not mean that it's safe in Duchenne, because, for instance, due to all the muscle turnover, the kidneys have a hard time in Duchenne patients anyway. So what may be tolerable for some patients or with another disease may not be tolerable for Duchenne. And of course, Duchenne patients use steroids, so there might be an interaction with that. So my last interactive example is about sildenafil. You all know sildenafil, this is Pfizer. And there was a study in MDX mice where they treated them with Viagra. And what this did, this reversed the cardiac dysfunction. So when they treated the mice, this reduced the amount of fibrosis in the heart in mouse. And of course, this was picked up by the media. But now my question to you is, knowing that this can reduce heart fibrosis in a mouse model, knowing that Viagra is already approved, would you prescribe it to your Duchenne patients Viagra? Or if you have a son or if you are a Duchenne patient, would you use it yourself? And again, it is anonymous, so don't be afraid to be honest about this. So I think it's about 50-50 between no and clinical trials. We have some people that are considering it. I will give you 10 more seconds and I'm almost gonna wrap up. So most people would wait or just not use it. Well, of course, a clinical trial was done, and the result of the trial was that the Viagra did not improve heart function in Duchenne and Becker patients. And this was the title of the paper, but if you dive into the data, what you will see is actually it made the heart function of Duchenne patients worse. So again, a mouse is not a human. A mouse has a heart rate of 600 beats per minute. It's different. So the fact that something works in a mouse doesn't mean it works in a human, and it can even be detrimental in humans. So this is why it's really good to wait for your clinical trials. So very important to not go too fast. So test one is, does it work? And then you need to optimize the dosing, the regimen, the route, etc., etc. If you go too quick to a clinical trial, these are the trials that are failed, where in the end you don't know whether the treatment really did not work, because maybe the dose was not optimal, maybe it was not safe, etc. So this is important to bear in mind. And there is the Treat NMD Advisory Committee for Therapeutics, and I'm aware of time, so I will not dwell on this too much, but you can go online to look into this treatnmd.org/tact. And this is a multidisciplinary team that can help clinical researchers or companies to develop their treatment and to de-risk it and to see, well, did you do all your preclinical studies that you should be doing? Finally, be aware that there is a bias to what works. So it's really difficult to publish about something that does not work or to see or to confirm that something that works really works. And this bias is in publications, but this bias is also there in social media. In social media, the parents who show that something works, they will be on social media. When things don't work, these are not the parents that will post this on social media. So take-home messages: scientists are humans. They may be over enthusiastic, so it's good to always do a reality check. Be critical when you hear news about Duchenne. What type of news is it? Is it a press release, a scientific publication, a news report? Is it an approval by a regulatory agency? If it's a publication, still be critical, because not everything is equally sound. And it's very important to don't hype, because families, patients, they need to make decisions on whether to take part in a trial or whether to have a treatment based on realistic expectations. So what can you expect from a treatment, and what are the side effects? And if you hype, then the expectations will be too high, and then of course you will accept burdens that are much higher than if you knew that the expectations would be much more limited. So thank you very much. I will stop sharing my screen, and I'm happy to take any questions that you like.